All right, we've come to our final chapter in the book. We're going to do chapter 25, the RNA viruses. And we're going to split this chapter up into 3 lectures. I've chosen 3 of the RNA viruses to present to you. The Orthomyxoviridae is a family of RNA viruses that has the orthomyxoviruses in it, and our orthomyxoviruses are the causative agent of influenza. Okay, so influenza, or what we would maybe just call the flu, that is not the same thing as stomach flu, right? Stomach flu is really a misnomer. It's better termed enterocolitis. Not very many people would say that, but enterocolitis or stomach flu obviously is the throwing up kind of flu. True influenza is a respiratory infection and rarely counts vomiting as one of its symptoms. All right, so when you get a flu shot, that does not protect you against stomach flu. All right, so what do we want to know? There are 3 types of influenza. Let's see. Make sure I didn't turn my volume off there. Okay, there are A, B, and C. Okay, influenza A is typically the worst in humans, most severe signs and symptoms. Influenza B, if you're infected with one of those viruses, it's clinically usually not as serious. It's very rare to have pandemics of influenza type B because it has a very limited host range, less likely to see novel types of influenza B, and we'll get to that shortly, a little bit better explanation of that. All right, and the 3rd type of influenza, I should say, that we see in humans is influenza C. It's fairly benign, it's a lot less common, and usually causes a very mild disease. All right, so if we look at the structure of the virus, all right, what do you want to know about that? It's enveloped. Okay, it actually has a helical capsid. This is a pretty nice schematic right here. You can see that helical capsid in there. It has a segmented genome. So again, some viruses have a linear genome, so all the DNA or RNA is just in one strand. But here we see either 7 or 8 segments. So there's 8 here, so I think influenza A and B have 8 segments, and C only has 7. Okay, and so each one of these segments encodes for 1 protein. All right, so if you remember with the herpes virus, those viruses typically carry about 80 genes, somewhere between 80 and 100. All right, so much smaller genome for RNA viruses. They tend to have smaller genomes. Okay, they don't have error-correcting mechanisms, so that's partly what accounts for that. It is a negative sense single-stranded RNA genome. All right, so they have to make their own enzyme, some kind of RNA polymerase that copies positive sense single-stranded RNA from the negative, because remember, our ribosomes only recognize that positive sense RNA. That's like messenger RNA. All right, let's look at some key structures of the virion. There are 2 types of spikes, of glycoprotein spikes. And those are important because those are the most antigenic part of the virus. That's the part that our immune system recognizes the best. H, or sometimes that's abbreviated HA, that stands for hemagglutinin. Okay, and in this picture here, those are the little red spikes. those hemagglutinin glycoproteins, we think, function in attachment of the virion to the host cell and fusion of the virion with the host cell membrane. Okay, and then the N up here, or sometimes it's abbreviated NA, that stands for neuraminidase. That's an R there. Okay, and that function is primarily for release of the virion from the host cell. Now, I've read that, and in the book, also it says that the neuraminidase can play a role in fusion of the virion with the host cell, with entry, but we think the primary function, again, is as it releases from that host cell. Okay, so the H's and the N's, we use the variations of those two proteins to name the strains of the virion. So for example, you may have heard of H1N1. We also call that the swine flu. But again, we name the strains based on the different variations of H. I think there's 10 different H's and 9 different N's. So you might, again, have H1N1 or H2N3, et cetera, et cetera. And some of those combinations don't infect humans. Some of them would maybe infect only pigs or pigs and birds or whatever. So when we make flu vaccines, we try to make the vaccines that recognize the different combinations or the different kinds of the spikes. Now, unfortunately, that's not easy because there's a lot of genetic variability in those glycoprotein spikes. They can change over time. Okay, so there's actually two different types of antigenic variation that goes on with influenza virus. So remember, we talked about another organism where that antigenetic variation is a problem for our immune system, a problem for us. That's why we can't make a vaccine to it. Remember, that was the Neisseria gonorrhoeae, as well as antigenic variation. That organism also has phase variation, remember, where the pili detach. Okay, so there's two kinds of antigenic variation that the influenza viruses can do. Antigenic drift, that's kind of a gradual change in the amino acid content or composition of those glycoprotein spikes. Okay, so kind of slow mutations over time. Remember that RNA polymerase doesn't have, I mentioned that in passing, doesn't have a proofreading function. So it makes a lot of mistakes. Okay, so basically those spikes can kind of stay ahead of memory cells. Okay, this is kind of a gradual change, though. It's not, every 10 days like we saw with the gonorrhea, but it can stay ahead of the memory cells in terms of kind of year to year. So we think antigenic drift is responsible for kind of every other year outbreaks of mild influenza, because it takes, gradually over a couple years for those spikes to change. So tend to have, 1 year's not so bad, the next year's a little worse, and then not so bad and a little bit worse. Again, that's because of antigenic drift. There's another kind of antigenic variation that happens only with type A influenza. And the reason is because type A influenza can infect other animals as well as humans. Okay, so this happens when the genomes from 2 or more influenza viruses infects a single cell. And then they kind of switch around a little bit. They reassort. Okay, so for example, if maybe you have a H3N2 virus that, say, is in a human, and an H5N1 from a bird infects a duck, for example, both of those in the duck at the same time, and they can reassort and make an H5N2. Okay, and this might result in some novel virus that really hasn't been seen before and nobody would have any immunity to. Okay, so we think this kind of abrupt antigenic shift is responsible for much more severe influenza, maybe about every 10 years or so. All right, so the last time we had really severe flu was the 2008-2009 pandemic, so we're kind of due for another bad flu year. Okay, and this next graphic kind of explains that antigenic shift a little bit, maybe better than what I did. So at the top here, we've got, here's a virus that's infecting humans. Here's maybe from a duck, maybe. And then if both of those infect, say, a pig at the same time, we can get some reassortment there, and a different form that now maybe can infect humans that hadn't previously. All right, and in China, the practice of living in very close quarters with your livestock is pretty common. Okay, we have maybe the pigs living in the house, even with the ducks and the chickens and whatever. So we think, this kind of reassortment is much more likely to happen in places like China. Okay, and then the antigenic drift is different. It's just kind of random point mutations over time. All right, as far as a little epidemiology, this shouldn't be anything that's too shocking here in terms of transmission. No surprises here. Primarily by large particle droplet. Okay, so sneezing, coughing, etc. We think also through fomites. And actually, for a membrane-bound virus, influenza is pretty hardy. It can be viable for about a day. Okay, so again, door handles, doorknobs, computer keyboards, that sort of thing, toys at the daycare, can also be airborne. Okay, so up to 6 feet. So remember, if it's more than a meter, then it can be transmitted further away than a meter, and then it can be considered airborne. Okay, so similar to coronavirus there. More likely to be transmitted in areas of large population. Okay, or densely populated. So back, say, World War I time, when we had troops moving all over the world and troops living in army camps, close together, the flu just spread like wildfire. And I want to talk a little bit about this 1918-1919 flu pandemic. If you ever get a chance to read in a little bit more in depth about this, it's really fascinating to think about and to learn about, especially in the age that we're in now. But it's estimated that about half the population of the world was infected. This 50 million mortality figure is probably on the low end. All right, so this flu was not the kind of flu that we typically would encounter now. You have a little coughing, a little fever, you're miserable for about a week. This flu was really special. It was very common for patients to bleed from the nose. You have hemorrhaging of mucous membranes of the nose. Pneumonia was really common. And actually, back then, they were quite good at treating pneumonia. Even though this was before the age of antibiotics, they could harvest antibodies from animals and other people, and they were very good at treating pneumonia. It's just when you had situations like this, where thousands of people were sick at a time, they didn't have the capacity to do that. It was also a hallmark of this flu, some neurological complications, so even psychoses, mental disturbances. Woodrow Wilson had the flu, obviously the president, and when he recovered, he started making decisions that were really contrary to how he'd acted before he was sick. And this probably impacted the treaty that we made with Germany. And, some historians say that that's one of the reasons that Germany was treated so kind of poorly and led to World War II. So it's really interesting when you think about how infectious disease, affects all sorts of, world events. Another interesting hallmark of this pandemic was that it was healthy people that were dying, people in their prime. Young adults were, for some reason, more likely to die. It had something to do probably with their robust immune response. So too much inflammation made them more vulnerable. And you would think a really severe flu like this would be caused by an abrupt shift. They think now that the evidence is actually that it was probably some kind of flu that at one point was avian and kind of gradually came to be able to infect humans and be passed in between humans as well. Okay, so another pandemic like this is inevitable, and this is something that really has people, that infectious disease specialists, really worried about it, because we're not much better equipped now than what we were then. We have a little bit better public health measures in place. We've got some drugs to treat flu, but they're really not very good. We do have better vaccine capacity, but our vaccine isn't super either. So it's definitely something to be worried about. And this picture is from your book there. They're filling up gymnasiums, when hospitals became overrun. Okay, so interesting to think about. All right, what does the clinical disease of flu look like? You probably all had it at one time or another, so again, shouldn't be any surprises here. But and then on an average, about 2 days after exposure is when a patient starts feeling the signs and symptoms. So about a 2-day incubation there. The virus binds to epithelial cells of the respiratory tract. Okay, and it kills those host cells. Basically, when the virions are leaving the host cell, they kind of leave it all at once. It's not really a lysis per se, but because they're endocytosing, they're taking some membrane with them, but it effectively has the same effect that lysing the host cell does. So it's killing the host cell and causes lots of inflammation, lots of cytokine release. Okay, so we see high fever as a result of those cytokine release, chills, muscle achiness, muscle pain, lethargy, cough, again, respiratory illness, lasts 7 to 10 days. Okay, and it can be even longer till the patient gets their strength totally back. Secondary infections are common. Typically things like pneumonia, that's particularly dangerous in elderly people, and otitis media or ear infections may be a little bit more common in children. Okay. So what weapons do we have to treat influenza? We do have a few antivirals that may help to shorten the duration of flu, especially if they're taken at the initial onset of symptoms. So typically they have to be taken within 1 to 2 days of getting symptoms. That can be difficult to do. You don't know how bad it's going to be. You need a prescription to take them as well. Okay, so most of the drugs in use right now are neuraminidase inhibitors. All right, so you may have heard of a few of these. There are 3 that are, I believe, approved for use right now. Relenza, I'll just get into the trade names. I won't get into their, the longer names. I don't have it right in front of me here, I guess. Relenza, Tamiflu, that's probably the most common one that you've heard of, and one called Pyramivir. Okay, so we'll get to how these are used in a minute. Let's talk about this in a minute. What are neuraminidase inhibitors? Okay, I found a nice graphic that explains this pretty nicely, and it kind of helps you understand how the virion is, what it's doing in our cell as well. All right, so let's start right here. Here's the influenza virus, or technically this is a virion, right, because it's outside of the cell. And we're interested in just these. This is kind of small here, but the hemagglutinin protein, that's the red one, and the little purple T's, that's the neuraminidase. All right, so for attachment, here it's showing the red hemagglutinin helping with attachment. And actually it fuses with the membrane, it gets endocytosed. So it's covered in this, encircled in this membrane, and it's carried through that membrane to the nucleus. Okay, ignore this over here. This is a class of drugs that we don't use anymore because it doesn't work anymore. Okay, so we've got that virion into the nucleus, and hopefully you're thinking, well, that's kind of strange. Why would an RNA virus need to go to the nucleus? It makes sense for a DNA virus like herpes because that's where the machinery is to replicate the DNA in the host cell's nucleus. But an RNA virus doesn't need the enzymes to make DNA, and we don't have enzymes to make RNA from RNA, so why go into the nucleus? Well, it's one of only 2 RNA viruses that does that, and we'll talk about why it does that in a minute. But they enter the nucleus and start synthesizing viral proteins. Remember, they need the ribosomes to do that. This doesn't, I guess this is supposed to be the rough ER over here. And those virions eventually leave the cell. And this is how it's typically going to happen over here, where the new virion buds off with the help of those little T-shaped neuraminidase proteins that helps this new virion pinch off and go on its merry way. Now, if the cell is in the presence of NAIs, neuraminidase inhibitors, that slows down the new virion from budding off of the host cell. It slows that down, and when it does bud off, the virions tend to stick to each other. We get virion clumping. Okay, so that's bad for the virion. Our immune system is more likely to notice antigens that are all clumped together. Okay, remember that's one of the functions of antibodies, to agglutinate. All right, so that, in theory, is what neuraminidase inhibitors should be doing, like Tamiflu and Relenza. Okay, how well do they work? Well, they actually, that's something that we argue about, and different studies find different things. Basically, we think they shorten the illness about a day. So minimal help at most. Anyone can take these, even children. It's recommended that people especially take one of these neuraminidase inhibitors if they're at high risk for complications. So anybody that's elderly or has some kind of underlying chronic issue, then the doctor is probably going to suggest that the patient take these. So again, you have to have a prescription, and they're about $100. So depending on your medical insurance or whatever, is it going to be worth it to shorten it by a day? There's also some side effects that are common, like nausea and vomiting, diarrhea, dizziness. So, this is a decision that you kind of have to weigh out, basically. Is it worth the side effects? Is it worth the money? Am I at risk? All those things have to be taken into account. So definitely educate yourself. Okay. The second, we have a new influenza drug that works by a novel mechanism, and you might have seen the commercials for this winter. It's called Xofluza, and actually that's a trade name. The other name is Baloxavir. Make sure I spell that right. So you might have seen the commercials for this. The flu sucks everything out of you. Those commercials. Okay, so this was new. It was approved in 2018, not 17, 2018. Okay, and so this mechanism, it says it inhibits cap snatching. Okay, well, so let me explain something that the orthomyxovirus does. The virus has to acquire a 5' cap from the host messenger RNA. Okay, so if you remember, if you go back to our chapter on microbial genetics, that was a difference between bacterial transcription and microbe and eukaryotic transcription. So in eukaryotes, they acquire this 5' cap to their messenger RNA transcript, and then it's that 5' cap that lines up at the ribosome just right in order to start translating that messenger RNA at just the right spot. Okay, so the virus for orthomyxovirus, this is why it goes into the nucleus in order to snatch that 5' cap from the host messenger RNA. Okay, and it uses it to actually as a primer to make its own messenger RNA. Okay, so it uses it to prime viral RNA synthesis as well, and then the messenger RNA can be read at the host ribosomes. Okay, so that's why the orthomyxovirus has to come down here into the nucleus in order to grab that 5' cap from the host messenger RNA. So this new drug, Xofluza, somehow inhibits that from happening. Okay, so that basically really slows down the replication of the genome of the virus. Okay, and this is just one dose. It can be taken orally, but only for people 12 years and older is what it's approved for. So other than one of those drugs, what else can we do just to kind of finish up here talking about flu? So continuing with treatment, basically bed rest, definitely until that fever breaks. You shouldn't be out, trying to do your job, passing it around to other people. Drink lots of fluids, some kind of non-aspirin pain relief. Okay, especially you don't want to give aspirin to children because of the risk of Rye syndrome, which is an inflammation of the brain. Okay. The only other thing is talking about the vaccine. So we touched a little bit on the flu vaccine before, and we were into chapter 17 talking about vaccines. Hopefully, in light of the current situation that we're in, hopefully if you've never had a flu shot before, you'll reconsider next fall. If a really nasty flu strain starts circulating, which again, this is what professional infectious disease people spend a lot of time worrying about. If you've had regular flu shots every year, year after year, you might have some immunity to, again, some really nasty strain that starts circulating and not get hit so hard, either not get sick at all, or maybe have a much more mild flu than what other people get. I just wanted to put up, this is the formulation for this year's flu vaccine. It got cut off of my last slide when I was talking about it before. This year's, there's lots of different variations of the flu vaccine. This one actually was quadrivalent, so it has 4 different strains in it. This PDM, that stands for pandemic-like virus. So this is very similar to that strain in 2018, what's considered the last, it was in 2008. But this is, again, it's H1N1. That was what the 2008, again, similar strain was that had the bad pandemic, again, about 10 years ago. So again, don't memorize that. That's just sort of interesting, the lineage of the viruses, where they were first isolated and the year. How good is the flu vaccine? It's not great. From year to year, it can reduce the chances of you getting the flu somewhere between 40 and 60%. And it varies, certain subtypes, it might vary from year to year. According to the CDC this year, our flu vaccine was 45% effective. You don't hear very much about flu now. We're concentrated on other things. In some years, when the vaccine hasn't been well matched, for some reason, some of the strains mutate. Remember, they have to decide on this 10 months ahead of time to give the vaccine makers time to produce enough. The effectiveness can be a lot worse if some of these strains mutate a little bit. It's very safe. There is about a 1 in 100,000 chance of the serious complication of Guillain-Barré syndrome. Now, that can be a complication of any viral infection that you get as well. But again, 1 out of 100,000 people get the flu shot can get this. It's a kind of a weakness and sensory loss due to the demyelination of peripheral nerves. Most people recover, but there are occasional fatalities to this. In 1976, there was a big swine flu scare epidemic, and there was a higher percentage of people that got this in 1976. So if your parents are old enough to remember that, then they may be a little bit biased against getting a flu shot, but it's much safer now. Vaccines are just getting safer and safer all the time. Okay, so this is a inactivated virus. Inactivated virus vaccine. It's typically injected, but they do have an attenuated or a weakened strain of the virus that's given nasally, and that seems to work best in children, right? So that works out well. They have kind of one less needle poke. Okay, so if you have questions about the flu or questions about the vaccine, I'd be glad to try to answer that or at least start a discussion. Our next video, we will talk about HIV. Have a good day.